Welcome, everyone, and thank you for joining us for the International Myeloma Foundation's Living Well webinar series. Today, we'll focus on new drugs in myeloma, what, how, and when. I'm Robin Toohey, Vice President, Support Groups. And before we get started, and on behalf of the IMF, I'd like to thank our sponsors for supporting this educational program, Bristol Myers Squibb, GlaxoSmithKline, Janssen, and Karyopharm Therapeutics. So before we get started, I'm excited to let you know that Myeloma Action Month has made a huge global impact. Through the IMF's Myeloma Action Month program, we've reached, drum roll please, Dr. Joe, this is a big number, 38 million people in almost 40 countries as of today. That is amazing. And that's because of all of your outstanding engagement. And together as a community, we've brought these stories forward that resonate and empower us. So thank you all so much. And just a reminder to all the support groups out there, the Support Group Wellness Challenge is still going on. You have about a day left to record all of your activities. It will be tallied by noon tomorrow on March 31st. So get those activities in, and that's going to be exciting to see how much all of the support groups across the United States have engaged. So now it's my absolute pleasure to introduce you to our speaker, Dr. Joseph Mikhael, who is the Chief Medical Officer of the IMF. He is also a professor at the Translational Genomics Research Institute. He did his medical training, hematology residency, and myeloma fellowship in Canada. He was a myeloma physician at Mayo Clinic in Arizona before joining the IMF in 2018. So Dr. Joe, without further ado, we are really looking forward to hearing your good information tonight.
Well, thank you so much, Robin. You're always gracious and so informative. And I'm very excited to be able to talk about this topic. It sounds a little daunting at first as we think about all sorts of new classes of drugs that we're going to be discussing together. But nonetheless, I think we can break it down, and I'll try to make it as straightforward as possible. You know, I always think at a talk, and in particular at these Living Well webinars, I try to provide what I call high-hanging and low-hanging fruit. So if you're here today and you really are new to myeloma and some of the words we're using are already unusual to you, no worries. We're going to try and make it straightforward. If you're here today and you kind of think that you're a veteran and you understand these things, hopefully I can give you some more information to think about and to understand. So really 5 things that I kind of want to achieve. The first is to review the key, what we call classes of drugs we use in myeloma. So although every drug is different and unique, we lump them in classes typically by how they work. And so what I'm going to do is the second objective is to give you an overview of how those drugs work. Now, you know, I could spend hours boring you with all of the basic science behind it and all the meticulous detail. I'm just going to try and make it straightforward because I think when we get a sense of how drugs work, we feel more engaged. One of the objectives overall really tonight is to have you engage more with your thinking about myeloma so that as you speak to your healthcare team, either as a patient or as a care partner or as someone who knows someone with myeloma, that you can understand more of the terminology that's being used. And then one area that I'm going to particularly focus on tonight is what we call immunotherapy, the concept of immunotherapy and what it means, because many of the new classes of drugs that we use in myeloma are under this umbrella of what we think of as immunotherapy. And the simple way to think about immunotherapy is that we are actually employing a patient's own immune system to fight their own tumor. And it's a bit complicated because myeloma really is a cancer of the immune system, if you will, where as I'll describe to you in a moment, certain cells that become cancerous are usually the ones that fight for you. And now, unfortunately, they're fighting against you. But one of the modern marvels of cancer treatment in many cancers, and in particular myeloma, is how we've employed other parts of the immune system to really fight someone's own cancer. And then we'll talk a little bit about drug development as how these drugs move their way from typically being tested where all other options have been used up, and how they make their way earlier on. And then in doing so, we're going to introduce some of the newest drugs and maybe a little preview of some drugs we don't have yet, but we know we're going to have in the very near future. So just to recap and make sure we're all level set here, I've already indicated that myeloma, if you will, is a cancer of the immune system. What do I mean by that? Sometimes we think of it as a blood cancer. What we mean by blood cancer is that it originates in the bone marrow. So inside of our bone is the factory of our blood. And our blood that we can do blood tests on is composed of different things. We've got cells that circulate in the blood. We've got proteins in the blood, and that's all made, if you will, in the factory of our blood in the bone marrow. And so in our bone marrow, we have cells, and whether you know it or not, you've actually been talking about these cells for the last 3 years. Every time you've talked about antibodies, when we talk about COVID and so on, we're talking about these very unique cells that make antibodies, and they're called plasma cells. So normal plasma cells are good. They make antibodies to fight off infections. So if Robin gets a vaccination tonight and she gets a tetanus shot, for example, or a COVID shot or a flu shot, or whatever it might be, that tetanus shot is speaking, if you will, to her plasma cells and saying, "Hey, Robin's plasma cells, this is what tetanus looks like. Please make antibodies so that if she steps on a rusty nail when she's out for a walk in not so sunny Connecticut, that her body can then react and see, oh, wait a minute, there's tetanus in the system." And they send out these antibodies to grab onto that tetanus and trigger the immune system to destroy it. So plasma cells are good. I've sometimes joked on these seminars that I want to get a picture of my wife's plasma cells and put them next to my bed, next to the picture of her. No, obviously I'm a little obsessed with plasma cells, but these are good cells that make good antibodies. Myeloma is a cancer of those plasma cells, and cancer means that it grows out of control. And so now these plasma cells that should be good now become bad, as it were, and they make bad antibodies that, instead of protecting you against infections, start to damage the body. They attack the kidneys, they attack the bones and other parts of the body. And so it's important for us to understand that basic principle as we go forward. And as I often say, it's not melanoma, which is a skin cancer. Myeloma is that blood or bone marrow cancer. Now, this picture looks so scary, and people look at this and say, "Dr. Joe, are you serious?" Well, I want to simplify this. When we talk about the bone marrow, we talk about the environment that these purple cells or these plasma cells live in. And one of the things that I want to impress on everybody tonight is really how complicated myeloma is. There's all these letters and numbers here. These are all sorts of different pathways that we've discovered as to how these cancer cells become cancerous and stay cancerous. All these different, we call pathways, are ways in which the cancer cell actually interacts with its environment. Because one of the things we've learned about cancer cells, it's not just that they grow by themselves and it's just, the tumor that sits there by itself, or these cancer cells. They interact with their environment. And it's that interaction with the environment actually that feeds them and they grow. And without going to a lot of detail, we have all these other cells in that environment, which include things like T cells and natural killer cells. And we're going to be talking about those cells later, because those are the very ones that I described before that we can employ to actually fight the tumor. So instead of those cells helping the tumor grow, we can now turn them on it so that we can destroy the myeloma. And what's fascinating here, of course, is that we have lots of opportunity to do that. Literally every one of these crazy letters on this really complicated picture is an opportunity to create a drug, what we call a target, that we can use to try and take down the multiple myeloma. And so again, making one more, I promise I won't have too many of these crazy pictures, but another one of these is, if you imagine that sort of green cell in the middle being the myeloma, it's got all these things that stick out of it. And sometimes we call those antigens. Interestingly, When we make antibodies, like the tetanus antibodies that Robin was making a few minutes ago when she got her tetanus shot, those antibodies, the way they recognize tetanus is typically they find something on the surface of the tetanus that they recognize and they hook onto it. That thing on the surface is called an antigen. And so we have all these antigens on the outside of myeloma cells, and that has really helped us, if you will, grab onto the myeloma. So think of it like hand-to-hand combat. It sees that, oh, there's something to grab onto this enemy, and they grab onto it. And so what we've learned over the years is that we've all these different targets, all these different antigens on the outside of the myeloma cell. And very often we use drugs or ways in which we can hook onto that antigen and trigger it, either, as I say, to directly kill the cell or to get other cells around it to come and kill it. Now, this again may look like a complicated table, but this is just an overview, and we're going to go into all of these over the next half hour or so. But we have all sorts of different classes of drugs. One of the classes that you may have heard about are sometimes what we call IMiDs, or immunomodulatory drugs. And what I've done is I've put the trade name first and then what we call the generic name second in brackets. It's really complicated sometimes because there's so many drug names, and people say, well, isn't Revlimid and lenalidomide the same drug? It is the same drug. And so every drug has what we call a generic name, and all of the IMiDs end in mide. But the trade names can be different, like Revlimid, thalidomide, orthalimid, and Pomalyst. Whereas proteasome inhibitors that we'll describe later, they all end in MIB. And so we have bortezomib or carfilzomib or ixazomib. If you go down to the bottom, we have monoclonal antibodies. I see one of them I switched by accident, but they all end in MAB. So there's daratumumab, elotuzumab, and isatuximab. So you got MIDs, MIBs, and MABs as the three big classes of drugs, what we call immunomodulatory drugs, proteasome inhibitors, and monoclonal antibodies. And then in the middle of the slide, we've added kind of the more old school drugs that we use, what we call chemotherapy, which are the more standard drugs that I'll explain in a moment. And then everybody's favorite, not favorite, which are steroids. So steroids are drugs that actually boost the effect of the others. So I'm going to walk through these classes with you in a minute, but we're not done. There are more classes of drugs. These are the ones that we sometimes think of a little bit more as more recent additions, where we have something called XPO1 inhibitors, sometimes called selective inhibitors of nuclear export. We have one drug in that class, selinexor. And then we have another two drugs that we had been using, and now the FDA has pulled from the market, although we still have some access to the second one. But I think it's important nonetheless to just explain them, because we may be seeing some of these drugs again, and both of them are still available in Europe. One's called a peptide drug conjugate, or melflufen. The other is an antibody drug conjugate, and I'll explain what that means in a minute, called belantamab or Blenrep. And then lastly, and what we're going to end really the talk with, is talking about these newest therapies that we really are excited about in multiple myeloma, what are called CAR T cell therapy, and I'll explain that process. And then most recently, literally just a few months ago, we had our first what's called bispecific antibody, or teclistamab or Tecvayli, that was approved. So we're going to talk through these. I know that's a lot to take in at once. So let's start with the basics. Well, what is chemotherapy really? And sometimes this is a word that we use very generally about almost any treatment we give in myeloma. Sometimes even if we're using some of these newer drugs, we sometimes use the word chemotherapy. But typically we use chemotherapy when we talk about drugs that tend to be a little bit more potent, in the sense that they hit all sorts of cells in the area. Unlike trying to just find that myeloma cell with some of these newer treatments, it kind of hits everything in the way. And so sometimes we see a little bit more side effects from this. And one of the oldest drugs we have in myeloma is melphalan, and actually still works well. We've been using it for 60 years. Sometimes we give it in high doses, like when we give people an autologous stem cell transplant, or sometimes called a bone marrow transplant, we use a high dose of melphalan. So what we do for that patient is we give them some kind of treatment, typically not with the melphalan yet. And then what we want to do is give them one big whopping dose of melphalan. I say to my patients, it's kind of like saying, well, you want to get rid of the weeds in your lawn. For those of you who live north, the green lawn will come back at some point, I know. I know it snowed this week in Connecticut, but poor Robin had to face that. But spring is coming, so you think to yourself, oh, I've got these weeds in the lawn, and so you put weed killer, as it were, on them. I know we have to be careful with that, of course, but just for the analogy, and that's kind of like giving people's initial induction treatment. But we can't really get rid of it all. So what you want to do is burn the lawn. Now, the problem with burning the lawn is it gets rid of the weeds, all right, but it leaves the good grass burnt as well. So what you want are seeds in the garage to be able to grow a new lawn. And that's kind of what we do with the stem cell transplant. What we do is we take seeds, or what we call stem cells, out of patients. These are cells that can grow a new bone marrow. Then we give them one big whopping dose of chemotherapy, and then we give them their stem cells back to grow a new bone marrow, to try and really get rid of what's left of their multiple myeloma after that initial treatment. And we still have evidence that this works. And then usually we give milder treatment afterwards. And so the drug we use for that process is called melphalan. Now, we often add almost to every drug class I'm going to be talking about today steroids, or typically dexamethasone. There are other forms of steroids. There's something called prednisone. There's prednisolone. There's different intravenous versions that we give to it, solumedrol. There's lots of different ways of talking about steroids, but they're really all part of the same family. Some are a little bit more potent than others. But I like to describe them like the booster rockets on the shuttle, like in this picture, because steroids are really good. They boost the effect of almost every treatment we use. What they do actually is quite interesting. It seems that they sort of interfere with the way tumor cells talk to each other. So tumor cells talk to each other, and that's kind of how they build themselves up. And this kind of interrupts some of that communication between them. So it's really helpful at first, but what we've also learned, like those booster rockets, is at some point they've got to fall off. You've got to have that dose dropping down. And what I sometimes call my favorite hashtag, down with Dex. And so we've come to learn that boosting benefit is really in the first few months, and we should be turning that volume down, as it were, reducing the dose of dexamethasone. And for most patients, we're eventually going to be in a situation where we actually discontinue the dexamethasone after a few months of therapy. All right, now let's get into these different classes that I described before. So we've talked about chemotherapy, we've talked about steroids. Now let's talk through the MIDES, the MIBs, and the MABs, starting with the MIDES. So the MIDES are lenalidomide, thalidomide, pomalidomide. And again, whenever there's this many pictures in the middle of it, it means that we don't fully understand what's going on. And I teach our medical students and residents, when a doctor's giving a talk and they shake their hands a lot like this, it usually means we don't really understand what's happening. So when it comes to describe these immunomodulatory drugs, although we have learned a lot more recently about something called cereblon and different ways in which this drug works, these drugs, we call them immunomodulatory drugs because they actually have a lot of effects on multiple pathways that I showed you earlier that affect the myeloma cell. So it's a little bit perplexing to fully understand how these drugs work, but they're very important in myeloma. And as you can see, they're given orally, they're given by mouth. And so they tend to be a little bit easier to take, although there are always challenges with every drug. And I should say that I'm not going to have time to go through every side effect and every detail of every drug. We're just trying to give people an overview today to get a feeling of how these drugs work. The second class are the proteasome inhibitors. And this was a drug, initially, the first one, bortezomib, that I had the pleasure of being a little bit of a part of as it was being developed even before it had a name. And it's really quite fascinating how this drug works. And again, I'll keep it simple, is that we have different things that get packaged and sent through the cell. And the cell has a different ways of breaking it down, different products that go through the cell. And proteasome inhibitors work in an interesting way where they inhibit what's called this proteasome, which is kind of one of these breakdown packaging plants for the cell, which tends to really be upregulated in cancer when cancer cells are getting really active and busy. And the proteasome inhibitors are very interesting in the way that they work. And now we have 3 of them that can inhibit the proteasome that we've demonstrated. All 3 of these drugs can really have quite an impact. Bortezomib or Velcade is given typically now in the skin as a subcutaneous injection. It used to be given intravenously, but now we try to give it more in the skin because it's has less side effects that way. Carfilzomib or Kyprolis is given intravenously, and Ninlaro or Ixazomib is an oral proteasome inhibitor. Now, as I mentioned before, as we now start to think about these other drug classes, not that immunomodulatory drugs and proteasome inhibitors aren't still critical. Oh, they're absolutely critical, and we use them every day in the myeloma clinic. Really, as we think of how the future of myeloma works, a lot of it, or how myeloma is treated, a lot of it has to do with your own immune system. And really, the first step into this or what's called monoclonal antibodies. So if you look to the left of this slide, we have those 2 Y-shaped things. One of them looks like it's grabbing onto something called CD38, and one looks like it's grabbing onto something called SLAMF7. Well, those are 2 things that stick out of the myeloma cell. I called them and described them earlier as antigens, or things that antibodies hook onto. So remember I told you that plasma cells make good antibodies to fight off infections. Well, that's such a cool phenomenon that we've actually tried to reproduce it in the lab. And so now we make antibodies, basically patterning after the way the body makes antibodies, which is a bit of a paradox. It's interesting, like you're trying to fight bad antibodies with good antibodies. It's sort of an interesting paradox in myeloma. But what we're doing is we now give a patient an antibody, much like those same antibodies that Robin made when she had her tetanus shot. And now these antibodies hook onto the myeloma cell at a very specific part of the cell, and they make that phone call, as it were, or trigger the immune system to destroy the cell. What's fascinating about this is that they're trained, of course, to hook onto that antigen that's on the myeloma cell. In this case, 2 of them are what's called CD38 or SLAMF7. But the good tissues around don't have that SLAMF7 or CD38. Now, we do have some cells that have a little bit of that, but it's really the first step towards the concept of a silver bullet or a smart bomb, as it were. Well, it's very specific to the cancer cells and not the good ones around it. And that's really what we ultimately want to do in myeloma, is when we give drugs, as I said before, like melphalan, that very good at wiping out the myeloma, like wiping out those weeds, but it also hits a lot of the good cells. Right? When people get high-dose melphalan, they lose their hair, they have a lot of damage to their GI system, and they get diarrhea and so on, because all sorts of cells get affected. But the concept here is to really just focus on hitting the bad cells and leaving the good cells behind. Now, there's another couple of drugs off to the right here that I'm going to come to later, something we call an antibody drug conjugate and a bispecific antibody, but we'll come back to that a little bit later. Let's stick with this monoclonal antibody concept. So again, what we're doing is we're giving the patient a drug that is a lot like a regular antibody that we can make, and it hooks onto the myeloma cell and triggers the immune system to destroy it. And it triggers the immune system through all sorts of different pathways. The immune system is, I mean, I know like I'm a hematology geek, but it's fascinating, all the different things that your immune system can do. And so this drug tries to kill the cell directly. It calls on different pathways. It actually calls on the body to make more antibodies to come and destroy it. It engages other cells, something called a natural killer cell. I mean, hey, if you're going to be immune system cell, I want to be the natural killer cell. I mean, come on, that's pretty impressive. And so these all these different mechanisms work to help destroy the myeloma cell. And we have 3 monoclonal antibodies. One's called daratumumab, and its target is CD38. We have one called elotuzumab, its target is SLAMF7. And then more recently, we've had introduced the isatuximab, which is also targeting CD38. And to give you just a little bit more of a picture here, you can see that there are different mechanisms in which both these sets of drugs work. But it's important to say that CD38 is pretty much expressed on all myeloma cells. So we see this sticking out of almost all myeloma cells, and very few other cells have it. Where some of our red cells may have it, some of our white cells have a little bit of it, but typically this is focused in the myeloma, and similarly to SLAM-F7, where we don't see that very much on other cells. All right, so we've talked about monoclonal antibodies. The other relatively new drug that we've just had for a few years now, called selinexor, is part of this family of drugs that are called XPO1 inhibitors. Now, and again, this is a fascinating picture, and there's lots of complexity here, but I'll make it simple. We have good things inside of our cell. In particular, the middle of our cells we think of, or the nucleus of the cell, the sort of the brain center of the cell. We have these things that sometimes we call them tumor suppressors. Tumor suppressor is a good thing, right? We like to suppress tumors. Well, what these proteins do in the cell is they make sure the cell is kept orderly. Remember, what was cancer? Cancer is when the cell is not orderly and it grows out of control, just does whatever it wants to do. These are kind of like supervisors here, right? Good supervisors. They keep things in check. They make sure that the cell does not become cancerous. But what happens, one of the reasons why cells do become cancerous, is they get rid of these supervisors. They send away these good tumor suppressors, and it goes out through sometimes what we call the XPO1 pathway. So we've learned in many cancers, and in particular myeloma, XPO1, these molecules that steal away the tumor suppressors and kick them out of the nucleus really get growing in the cell, and there's lots of them. So what this drug does is it inhibits that pathway. It stops that pathway, so those good tumor suppressors stay inside the nucleus, do their job, and the cell is less likely to be cancerous. Now, I've kind of oversimplified it a little bit, but it really is a fascinating pathway. Some of you who are, giving some high-hanging fruit here, some of you are familiar that with one of the problems we see in myeloma is where people have a deletion or a removal, if you will, of part of chromosome 17, what we call the 17p deletion. That's a problem because on that part of a chromosome, we make a really important gene, what's called the p53 gene. Well, p53 is one of these tumor suppressors. So when patients don't have that p53, it's almost as if they've, left the cell. They don't have those supervisors to keep the cell under control, and so the cell can become cancerous. And so this is a drug that we use not only in patients that have that chromosomal abnormality, but we can use it in all myeloma patients in relapse now because it works by blocking that XPO1 so that those tumor suppressors can stay within the nucleus. Now, this is a drug that, as I mentioned before, has been pulled from the market, melphalan flufenamide, and so we don't see it as much here, although it is still active in Europe, and why we included it in tonight. And this is kind of an interesting phenomenon where, remember, I shared with you what melphalan was before, but one of the problems of giving that high-powered melphalan or big dose of melphalan is that it hit not only the good cells, it hit not only the bad cells, it hit a lot of good cells too. And it wasn't, if you will, specific to the myeloma cells. Where here, this is a drug that's been if you will, chaperoned by a peptide, so by a protein that's going to be able to bring this drug into the cell and then drop the melphalan off, as it were, so it's very precise to only hit the inside of that cell. I kind of think of it like an Uber driver, right? The Uber driver picks up the melphalan and brings it specifically to its destination, and so it doesn't end up going elsewhere. And this is the drug that's given intravenously, melphalan flufenamide, which, as we mentioned, is not currently available in the United States, but is in some other jurisdictions. The other drug that was recently pulled from the U.S. market, although still available to some patients through a compassionate use program, I still have several of my patients on it and still being studied for potential use in the future, is Blenrep or belantamab. And we call this drug an antibody-drug conjugate. So remember how I said a monoclonal antibody is a drug that comes and hooks onto the outside of the myeloma cell and then kind of makes a phone call and triggers the immune system to destroy the cell. This is the same concept, but with one additional twist. is that attached to the cell is a toxin, if you will, or sorry, attached to the drug is a toxin or is something that can be very damaging to the cell, so that when the antibody hooks onto the cell, it gets engulfed into the cell and it releases this toxin to sort of destroy it right away. So it not only calls on the rest of the immune system to destroy the cell, it itself has a damaging presence to the myeloma cell. And that's the drug belantamab or Blenrep. Now, as we come to the last two classes, because I really do want to leave a lot of questions, time for questions, and I will remind you, as Robin reminded us at the start, that please feel free to submit your questions. I see lots of them are already coming in, which is great, and I'm more than happy to try and answer all of them, or as many of them as we have time to. As we get to these last two classes, these are really the newest drug classes in multiple myeloma, and I want to spend a couple of minutes explaining them to you because these are really unique and quite exciting. And this picture may seem a little complex at first, but let's walk through it. The concept of CAR T-cell therapy is basically to employ a patient's own T cells to fight off their myeloma. Remember, we've talked about immunotherapy. So immunotherapy is a concept where we take a patient's own immune system to fight off their cancer. And this is really a fancy way of doing it. I mean, if you told me about this when I was in medical school, I thought you'd be like talking about some Star Trek movie or something, because it really is so futuristic. And just to remind us that the T cells, we have different cells in the body that kind of function like soldiers. They can be trained and they can help fight. Of which one of the most common are T cells. They're called T cells in case you want to win with a Trivial Pursuit game next week at their family get together. They're called T cells because they actually originated in your thymus when you were very, very small. We also have something called B cells, and they're called B because they originate in the bone marrow. But then we have other cells, as I showed you earlier. There's a natural killer cell. But the concept is similar, and now we're working with T cells, although we are starting to work with those other cells as well for CAR therapy. And it's called CAR because it's what we call chimeric antigen receptor, and I'll explain that in just a minute. So basically, we want to do is we want to take T cells from a patient, and we want to train them to know exactly what that patient's myeloma looks like, and then give them back to the patient to destroy their myeloma. So the steps that we take, number one, is we collect the T cells from patients. For those of you who may have gone through an autologous stem cell transplant where we collected your stem cells, it's quite similar. People go through what's called apheresis, where those cells are collected. And then in the lab, which is what's in the purple box here, everything up until then is done in the lab, is that we start to, if you will, to train those T cells to know what the myeloma looks like. And how do we do that? It's pretty complicated, but pretty amazing. Two major things we do. We actually can multiply these cells. So I can take 100 T cells out of a patient and make 1 million, which is really remarkable. But what we do is we get those T cells on the surface of the T cell to express a receptor that is going to be specific to that patient's multiple myeloma. And that's why it's called CAR, or chimeric antigen receptor. So now you've got this T cell that's got that, what looks like an antibody, if you see the picture, sticking out of it, that is waiting to hone on to that myeloma cell. And that can be by different targets. One of the most important targets on a myeloma cell, I showed you earlier, CD38, SLAM-F7, we now very much employ a relatively newly understood target called BCMA, or B cell maturation antigen. And so as we produce these CAR T cells in the lab, it does take some time. It can take sometimes as little as 4 weeks and sometimes as much as 8 weeks. There's great research going on to try and even shorten that to a few days, which would be quite remarkable. But nonetheless, about a month or so later, we now have all these activated, ready to go to battle T cells that know what this patient's myeloma looks like. and then we infuse them back in the patient. Typically, a few days before we do that, the patient gets some form of traditional chemotherapy so that their own T cells don't fight with the new T cells you're giving back to them. And so then they receive those T cells. And that's the process of CAR T cell therapy. So in summary, we're taking T cells out, we're training them to recognize the myeloma, we're multiplying them in the lab, and then we give them back to the patient. Really quite remarkable how we are able to do that. And so just to give you a little picture here, so there's the myeloma cell on the left, is sort of a pinkish color, and there's that CAR T in green with those purple projections with having that CAR on it, or their chimeric antigen receptor on the outside of it, that's going to recognize that myeloma cell to go and destroy it. And right now we have two forms of CAR T cell therapy, one drug called Abecma, which is idaptogene viclucil, we call it ide-cel for short, and then Carvykti or ciltacabtagene autoleucel, which we shorten to cilta-cel. These are both given intravenously and typically require some time in hospital. Some patients may not need to stay in very long, but there are some, as I mentioned, I don't have time to speak about complications tonight, and side effects, but some patients can experience some challenges with this, although we have seen unprecedented response rates with CAR T cell therapy. Some of our clinical trials were over 95% of patients responded to this form of therapy. And then the last kind of treatment or class of drug I'm going to mention tonight are what we call bispecific antibodies. So if you listen to what I said earlier about monoclonal antibodies, and you just heard a little bit about CAR T cell therapy, it's kind of like bringing these two things together. So they're called bispecific because they have two arms. One arm, in the traditional monoclonal antibody way, hooks onto the myeloma cell, but now it has a second arm that reaches out to a local T cell. So instead of having to collect T cells and manufacture them and reinfuse them in the patient, we just get this drug off the shelf, give it to the patient, and it reaches out to that local T cell and, if you will, engages it, and says, hey, T cell, here's a myeloma cell, go to it, go destroy it. And it's really quite amazing. Again, almost sounds like a Star Trek movie or some kind of science fiction, that we can actually give a drug that holds onto the myeloma cell and hooks onto a T cell, brings the two of them together to destroy the cell. And that's the drug teclistamab or Tecvayli, which we now, just as I've mentioned, have had recently approved. But just to give you a flavor, looking to the very near future, we have another drug. And by the way, I don't make all these names, but I mentioned so far that the CAR T cell therapy we use and the bispecific therapy we use target BCMA on the surface of a myeloma cell. Well, we have a new target now, something called GPRC5D. They all sound like license plates. I don't know how that happened, and we have a drug similar in some ways to teclistamab that has one arm that hooks onto the myeloma cell and one arm that grabs onto the T cell. But now, instead of grabbing the BCMA on the myeloma cell, it grabs something called GPRC5D. And similarly, we have yet a third bispecific antibody class where we're not targeting BCMA or GPRC5D. We've got another license plate for you. We're targeting FcRH5. And this is the drug cevostamab, where similarly, this drug can hook onto the FcRH5 in the myeloma cell and with the other arm engage a local T cell. And before I wrap up, there are a whole host of new classes of drugs. I mean, I could bore you, Robin, and the team for hours talking about all these new drugs. But one that I want to highlight is a class of drugs that are now being called CELMoDs, of which we have two, one called iberdomide and one called mezigdomide, which are really kind of the next generation of those immunomodulatory drugs that we've called before. You remember we had thalidomide, lenalidomide, pomalidomide. Well, these also end in mide, although we think of them as a bit of a different class. And it's really quite remarkable. We're seeing them work even in patients whose disease has grown on those previous kind of drugs. Lastly, we have a drug called modakafusp, which is really quite interesting, where it's taking the same concept of a monoclonal antibody that's directed against CD38, so a little bit like daratumumab and isatuximab that we showed you before, but now it's got hooked onto it a really old school drug. Now, most people listening probably don't remember this old drug that we gave over 20. When I started in myeloma 25 years ago, we were really coming to the end of using it, but a drug called interferon. But now, a little bit like the concept I shared earlier with melflufen or melflufen, we can now, in a more precise way, bring this old school drug right into the cell through a drug called modakafusp. And then there is a whole separate class of drugs that we could almost devote a whole other hour to. But we do know that there is a form of myeloma where patients have a genetic abnormality called T1114. It just means that a piece of chromosome 11, a piece of chromosome 14 are brought together, and it activates a pathway that keeps the cell alive and keeps the malignancy going. And we can block that pathway with a drug called venetoclax. It's not yet FDA approved for this indication. We use it in leukemia, but we're likely going to have access to it in the near future. And really, there is more to come, as I've said. We're looking at different ways of giving CAR T-cell therapies where maybe we don't have to do all that collection. Maybe we can just build CAR Ts in the lab and give them to a patient. These bispecific drugs, we're looking at engaging T cells now, but we're looking to engage even other cells in the future. I have a clinical trial open now, for example, where we engage natural killer cells, where patients may not experience some of the side effects that they would from other drugs. And so many other drugs are being developed. And I'll just close off with, trying to, for those who are conceptually thinking that there's all these different ways now, going back even to what we described at the very start, lots of new drugs and new ways to affect multiple myeloma. And I will say that typically, as we develop these drugs, it reminds us, and as we discuss these drugs, how important myeloma research is. These drugs don't just get discovered overnight. These take years and years of development, and we work in the lab. We do animal studies, so we do clinical trials, what's typically called phase 1 trials, where they're tested to see how safe they are, and then phase 2 and phase 3 trials, where we test them to see how effective they are and how they compare to the standard of care. And typically, because a lot of people say to me, well, Dr. Joe, this CAR T-cell therapy looks so great. Why can't we just use it right off the start? Well, drugs have to go through a certain development. We need to prove their principle in an area of what we sometimes call unmet need, where everything else has failed, because we want to be safe. If you ever work with me in my clinic, I say it all the time, safety is paramount. We want to make sure that it's safe, and we use it in that context and eventually see it move forward. Examples of this are daratumumab. When daratumumab was first released, we used it only in patients that had just about everything else, and now we use it frontline. Similarly, with CAR T-cell therapy, we're using it now after patients had at least 4 lines of previous therapy, but we're now bringing it earlier, and eventually I think we may even see CAR T-cell therapy up front and even potentially replacing stem cell transplant in some patients. Well, I've tried to just give you a little bit of an overview. Oh, there's so much more that we could talk about, but I am going to leave you here with lots of other resources. Robin reminded us, go to myeloma.org. You can go there, and I've shown some subtitles here, sub pages within our page, where I've written some articles here. Dr. Dury has written some articles. Many other things are available to have you understand these drugs a little bit more as we go forward. Now, let's turn to the Q&A. I hope to leave 15 to 20 minutes, and we're exactly 17 minutes left. And wow, Robin, the questions are pouring in. I see 30 of them already, so I'm going to take a deep breath, and I'm going to be ready to try and answer some of them. So here we go. Question number 1. If you become refractory, so refractory means that your disease grows while you're on a drug. If you become refractory to Revlimid, can you introduce it again later after some time passes? Thank you for this great question. In short, we typically don't use it right away because the disease right then has grown on it. But we know that sometimes the disease can change over time. So sometimes we do go back to drugs that the patient's been refractory before. We typically don't do that until they've seen most of the other drugs and different mechanisms of action. That's actually one of the reasons why we gave this talk tonight on different mechanisms. So you could think about how we come at myeloma in all sorts of different ways, but it sometimes can be. Are there specific treatment clinical trials, research for translocation 414? So I mentioned earlier one form of myeloma called translocation 1114. When we say translocation 414, let me just remind us again, it just means that typically a piece of chromosome 4 that should be on chromosome 4 and a piece of chromosome 14 that should be on 14 get together, and these two bits come together. Unfortunately, with translocation 414, we typically think of that as so-called a high-risk myeloma, which means when that's present, the disease is a bit more aggressive. It tends to grow more quickly, and it tends to be more difficult to treat. We do not currently have a very specific treatment for translocation 414, but I have to say different drugs are being explored. There's a pathway, kind of like I said with translocation 1114, the BCL2 pathway gets upregulated. With translocation 414, it's a different pathway called FGFR3 that gets upregulated. And so we are trying to develop drugs specific to that pathway. Can you explain the function of the new Revlimid? I think you're probably asking about that class of drugs called cell mods that I mentioned a picture of earlier. So I think this question may have come in before I fully described them. But these are drugs that work in a similar way and a similar part of that cereblon molecule that I mentioned before, but actually seem to be able to work even in patients that have previously had Revlimid and pomalidomide. What are the after effects of daratumumab? Again, that could be a long answer, but short. Daratumumab, that monoclonal antibody, and the other one similar to it, isatuximab and even elotuzumab, typically the greatest side effect we see is when they first get it, that the body can react and have what's called an infusional or administration reaction. Thankfully, we can manage those really quite well now with the different treatments we have and preventions that we have. That's why we give patients a bit of steroids and Tylenol and a few things before they get it. Down the effect, there can be a few others. It does suppress the immune system to some degree because in daratumumab, for example, CD38 is also expressed on some relatively normal plasma cells. And so people's regular antibody response may not be as aggressive as we learned a little bit, unfortunately, during COVID. And it can reduce people's white blood cell counts a little bit. Those are typically what we see a bit later. You just referred to antigens that stick out of bad plasma cells. I think I've heard them referred to as proteins. Are both terms correct? Yes, we try to use the word antigen because then it's specific to that kind of protein. A lot of the things we've described today have a protein element to them because there's so much of our bodies and cells that have proteins involved. But typically we try to use the word antigen because then you specifically think of something that's sticking out of a myeloma cell. Why did the FDA remove the 2 myeloma drugs that are still available in Europe? That's a great question and a lengthy question. You know, it's tricky. You know, these drugs, we always want safety above all, and drugs have a pathway to approval now where if they can prove that they're active, even just on their own, then there's something we call accelerated approval. But then that has to be more validated later in a randomized trial where a drug is compared to another. And sometimes those randomized trials don't fully validate the initial signal. And that's really essentially what happened with these 2 drugs. That being said, there are other studies that are being done to try and demonstrate the efficacy of those drugs. So we may be able to see them again. I just can't be a prophet and predict the future. Can you talk about inflammation versus myeloma bone? Yeah, that's a complicated point because, you know, the inflammation pathway and the immune system are very much connected. And I think what maybe the questioner is seeking here is to try and understand what's happening in the bone itself. And so those myeloma cells, they're active in the bone. As I said, they nourish themselves from the things within the bone. This is one of the reasons why bone get thinned, and sometimes we can have holes in the bones that break. And so one of the things we can do to try and strengthen people's bones is not just to treat the myeloma, but also to give them bone strengtheners. I purposely didn't talk about those tonight only because I can only cover so many drug classes, but that's one of the things that we do. Any thoughts about a cure? Where are we on this? Well, I love that kind of question. you know, the ultimate mission of the IMF is to seek for a cure, and we want to find a cure. And until we find a cure, we want to support our patients and their care partners through their journey with multiple myeloma. Dr. Dury leads our Black Swan Research Initiative, which is really designed to find a cure. And you know, the way I describe it sometimes is that, you know, we're not just going to wake up one morning and have a cure to cancer, right? It's step-by-step process. We haven't cured diabetes. We haven't cured high blood pressure. We control them in the long term. All I can say is that we are so much further ahead now than we were even 20, 25 years ago. I know when I started, let alone 5 years ago. In fact, I could argue that this may be the most exciting year of myeloma research I've ever seen in terms of the number of drugs and the number of things that we're learning about the disease. I have had multiple fractures, even using Zometa. Why aren't other bone-building medicines, like some others listed here, eventually used? Yeah, that's a great question, and obviously one you have to discuss a little bit more with your provider. But we are trying to understand this a little bit better. There's some people that just don't respond as well to the bone-strengthening drugs we have. I think the principle I shared earlier is important, that the best way to rebuild a patient's bone actually is to control their myeloma while giving them a bone strengthener drug. And so we are looking at ways of giving other drugs. And there are other ones now. We have another FDA-approved drug, denosumab, that we use for myeloma patients. If selinexor is inhibiting the myeloma from getting rid of tumor suppressors, do the tumor suppressors that have already been exported regenerate? Oh, wow. Someone's really thinking about this, which is great. I mean, these are constantly being made, yes, in many respects. So it's not like once you've lost that tumor suppressor, like that P53, it's gone forever. There is regeneration. Hi, Dr. Joe. Do you think at some point stem cell transplant will be obsolete and CAR T will be the treatment of choice? I have to be careful because I have friends, very close friends, that are bone marrow transplanters. No, we want, of course, what's best for the patient. I can say that we are doing clinical trials now where patients are being randomized to compare transplant to CAR T-cell therapy. And what I've learned is don't be a prophet, Dr. Joe, just be Dr. Joe. I can't predict the future. I suspect that there will be some patients who historically we may have done transplant in whom we may do CAR T-cell therapy instead. But we just need more data to prove that. Please explain the function of the lambda and kappa light chains in relation to myeloma. Again, this is a great question. Lots we can say here, but I simplified things when I said myeloma is a cancer of the plasma cells that make antibodies. Antibodies are those Y-shaped things that we've seen on several pictures tonight. Interestingly, they're composed of two bits. There's a longer chain that's heavier, we call the heavy chain, and then there are little light chains on the side. Most patients produce both heavy and light chains, but there are about 20% of patients that only produce light chains. And so there are two types of light chains. Some are lambda and some are kappa. Normally, our usual plasma cells in a non-myeloma setting should have an even distribution of lambda and kappa. But when we have cancer, which is identical uncontrolled growth, one of those two dominate. And so very often in a patient, we're following their lambda light chain level, or another patient, their kappa light chain level, to understand their disease. Today is the only treatment that might create a cure CAR T, since the manufactured T cells will remain in the body and continue to destroy the myeloma cells forever without any special drugs. The other treatments and their side effects will continue to be needed to destroy to keep the level of myeloma cells low. What is the state of research that is focused on stopping the myeloma cell production in the bone marrow? Great question. You know, one of the benefits of CAR T-cell therapy right now is we give this treatment, and then we typically don't give treatment right after. People get my favorite drug, nothing, for a period of time after their CAR T. But despite the amazing response rates we're seeing, and sometimes it lasting a long time, CAR T in its current state is not really a cure, because nearly all of those patients, unfortunately, will relapse later. So we need to continue to do research and understand, is it CAR T plus something else? Is it a better form of CAR T? And we are working what I call CAR T-cell therapy 202, right, where we're getting better at doing these things. There's no drug we use in myeloma now that we use exactly the way it was first introduced. We always make things better to increase their benefit or efficacy and to reduce their toxicity or their side effects. And I think that's going to happen with CAR T-cell therapy. Someone else asked the same question as before, why were the drugs taken off the market? Does Blenrep attach to BCMA? If so, if a patient is considering BCMA-directed CAR T, should they avoid or postpone Blenrep? It's a great question. We're doing a lot of work now trying to understand this notion of, can you go from one BCMA drug to another, like from the antibody drug conjugate to CAR T, or from CAR T to bispecific, or vice versa in any of those situations? I think the short answer is it may not be optimal, but sometimes it's the only choice we have, but that there is still benefit. We had a recent publication from the group at UPenn. Adam Cohen led it, a great study looking at people that had another BCMA, including belantamab, and then went to CAR T. It was still very effective, maybe not as effective as we saw in patients that had not seen BCMA before, but still remarkably effective. Is it preferable to do a CAR T therapy before bispecific? That's the same principal question. Great question. We're still trying to sort that out. What is the best sequence? And that's going to take us time to figure that out. Right now, unfortunately, there's a backlog in trying to get people any of these drugs, to be honest. It's not that easy to get a CAR T or a bispecific. We hope that the supply is going to increase. So sometimes I say, the best one is the one you have access to, right? So right now, we don't know exactly the answer to that question. Can you tell us about a drug called Venclexta? Well, that is the venetoclax that I mentioned that we can potentially use in patients with translocation 11-14. There's another question, the same as before, about CAR T and frontline therapy. How long does CAR T last versus stem cell transplant? It really depends on the patient. And you have to remember right now, stem cell transplant with maintenance therapy, on average, it lasts about 4 years. In CAR T, we have to recognize that it's a very different patient population. In transplant, we typically give in patients first line of therapy after their induction treatment, whereas CAR T we're using many, many lines later. In the first CAR T cell product on the trials with ide-cel, we saw an average about 9 months, but it's over 2 years now with cilta-cel, the second product. So it's hard to compare those two that way. That's why we need to do those clinical trials. Robin, I know I've got 3 minutes left. I'm trying to catch up here. We're down to question 20, which I think is good. We're catching up. Are there any studies that show that repeated COVID boosters do no harm to myeloma patients? Does it not reignite myeloma? I think what we learned through the pandemic was that we really do not see myeloma worsened by boosters or by having a vaccine. If anything, it provided a tremendous amount of benefit for most of our patients. Not everybody, of course, had full benefit from it. We understand that, but we really don't see it worsening the myeloma. Okay, there's a couple of very sort of specific questions about someone's treatment, so it's going to be a little bit harder for me to answer that. If it's okay, Robin, I'll take maybe a couple more questions and then turn it back to you. We'll try to get as many of these answered in the future as possible. But here's a question. What is your biggest concern or frustration or challenge with myeloma progress? And what can we do as patients or consumers to help you and others who are experiencing the same thing? What a great question. Maybe this is the question that we're going to end on, only because I just can't get to the rest of the now up to 46 questions. I'll tell you what frustrates me the most is that we still lose patients to this disease. My team walking out of a room with a patient will often hear me say, I hate myeloma. And I do, because I see the impact it has on beautiful and lovely people that I care for. I say it all the time. I don't treat myeloma. I treat people. And so the hardest part is to see that even with all these great therapies, and even though we're seeing most of our patients live a long time, often with actually quite a good quality of life, we still lose in this country about 13,000 people a year to myeloma. It's terrible. Furthermore, as some of you know, I do a lot of work in health disparities. And it's terrible to me that there are people based on their race or ethnicity or socioeconomic status who have a delayed diagnosis, who don't have access to therapies. And what can we do? Well, join us in the IMF in the work that we're trying. Join a support group. Learn more at myeloma.org. If you have capacity, support us in any which way you can. And we are so grateful because we want to better the research, improve the diagnosis, improve access, find a cure so that we can support patients and their care partners and their families through what is often a terrible journey. We've come a long way in myeloma, but we're humbled on a regular basis of the magnitude of this disease. And that's why so many of us have committed so much of our lives to taking down this disease. A beautiful question to ask, and I hope it inspires us to do more. Robin, I'll turn it back to you to close off.
Sure. That was an absolute beautiful way and an important way to end the program, because as far as we've come, we're not at the cure yet, and we don't want to lose any more friends and family members. And so listening today to your explanation of what is a very complex disease, it helped to simplify but give hope, because this explosion in drug development in myeloma over the last several years, it's significantly improved the lives of patients with myeloma. I did see a question that you had about quality of life, which now we know, right, Dr. Joe, is included in clinical trials, which never used to be. So we have come such a long way. And it's why it's important for myeloma patients and care partners to learn as much as they can. And this is Myeloma Action Month. Become engaged, share your story, have key conversations with your healthcare team, because we know shared decision-making is a team effort. So we all can get engaged with this. So thank you so much, Dr. Joe. And thank you, of course, for all these really smart questions. I'm so grateful that the people involved with myeloma have really engaged to learn and improve their own quality of life just by hearing from people like you, Dr. Joe. And on Doctors Day, to all doctors out there, thank you for what you do. And thank you, Dr. Joe. And of course, again, we do thank our sponsors, Bristol Myers Squibb, GlaxoSmithKline, Janssen, and Karyopharm Therapeutics, for supporting these educational programs. And we just want to remind all of you that we are going to have this video replay available. It'll be up on the IMF website, so you can go back and replay it. I'm sure you're going to listen to it again and hear some different things. And I know those support groups are going to want to replay this at their meetings so they can pause it, stop it, have some good conversations. And again, your feedback is what we listen to. It's what drives us to create better programs for you so that they're valuable and beneficial for what you need to know. So thank you for taking your time tonight. Please make sure you take this survey, provide us with your feedback so we can continue to do what we do for you. And we thank you all for being here and sharing your time. Thank you, Dr. Joe.
Thank you.